So before we get to the speaker talk, I just want to give a brief overview or introduction of PacBio technology for rare disease research. For those of you who are less familiar with long reads, consensus hi-fi reads are the basis of PacBio smart sequencing, and it's been shown that our hi-fi reads are both highly accurate, as shown on the left, with around Q33 accuracy, as well as long. So in fact, hi-fi reads can be up to 100 times longer than short read NGS. So long and accurate reads with hi-fi enable a more complete view of the human genome. And at a germline DNA level, this allows us to capture not only small variants and indels, which short read whole genomes can adequately capture, but also allows us to get numerous other types of variation, including structural variants, repeats, and pseudogenes. And with PacBio technology, you can also get full phasing and the methylome as well. It's been shown that structural variants alter significantly more nucleotides in the genome than the S and Vs that can be captured with short read. And about 50% of the structural variants in ClinVar are actually pathogenic. So you're really getting a much more comprehensive view of the genome if you're using hi-fi long read sequencing, which can be critical in trying to understand rare disease. There are several examples of researchers who are already using PacBio sequencing for rare disease. These two studies just highlight the increase, for example, in structural variants and de novo mutations found just by looking at germline DNA with hi-fi sequencing. In this case, both of these led to an increase in explanation rate when they were looking at rare disease cases. However, it's becoming increasingly more apparent that not all rare disease causal variants can be found in germline DNA, and some that are found are of unknown function or significance. So we have more studies that are looking beyond DNA to include transcriptomics, for example. Studies of short-read RNA sequencing, which has some limitations that I'm going to discuss in the next slide, have already been shown to improve solve rates as much as 36% compared to just short-read exome or genome sequencing alone. And given the improved full-length transcript information you get with long reads, as you'll see in our talk today, we think long-read RNA sequencing is an even more powerful tool for rare disease research. This is because isoforms and not genes can be the drivers of disease, and variants in RNA can lead to disrupted expression, function, or stability that might explain mechanisms of rare disease. The graph on the right just shows that there are multiple classes and a high proportion of variants that can only be detected with RNA sequencing in a cohort of suspected Mendelian disorder samples. So why would we use long reads for RNA sequencing? With short read RNA-seq, the cDNA are fragmented. These are represented by the little light gray bars on the bottom. And then they're assembled computationally. And this is a problem because alternative splicing produces many isoforms that look similar, making it impossible or difficult to unambiguously infer the original set of transcript isoforms. In this schematic, those are the colorful bars shown above. In contrast, the PacBio iso-seq method sequences the full-length cDNA up to 10 kb with no assembly required, providing a complete view of the transcriptome at the isoform level. So we recently announced Kinnex kits for full-length and single-cell RNA sequencing, which uses the iso-seq method I mentioned, but further concatenates transcripts on a single hi-fi read for full-length isoform resolution at a higher throughput. In the top left figure here, we show that Kinnex full-length kits can capture long transcripts across a variety of species and tissues. In the bottom left, we show that you can get up to 16 times more data compared to our previous iso-seq method when you use Kinnex kits on Sequel IIe and even more on Revio. And then finally, with the increase in reads, we are starting to saturate known isoform detection. So the middle graph shows roughly 80% of known isoforms are detected with just 10 million reads across different samples. That's where this dotted gray line is and where the peaks kind of plateau. 10 million reads is the expected throughput you would get for multiplexing 4 samples on a Revio SMRT Cell, or you can get about 40 million reads for 1 sample per Revio SMRT Cell. So this increase in throughput allows you to capture isoform abundance, detect novel RNA variants, and characterize differential transcript usage. And if you'd like to learn more about the technical details of Kinnex and the workflow and analysis, please check out this app note. I've added the QR code, and this app note for the full-length Kinnex kits as well as the single-cell Kinnex kits are available in the handouts module. And if you want to try things out, we do have a promo around the Kinnex kits, so please reach out to your local PacBio rep. So with these kits, you have a full toolbox for tackling rare disease cases using genomics, transcriptomics, and even beyond that, all on the Revio platform. And to wrap up, here are just 2 example publications where full-length isoform sequencing with PacBio helped identify disease-associated variants in rare disease cases. So on the left, the authors looked at an undiagnosed disease network proband with a variant of unknown significance in a candidate gene. And this branch point sequence variant was actually found to produce 6 major transcripts, which they were able to identify because of full-length isoform sequencing with PacBio. And they found that 5 of the 6 transcripts had disrupted open reading frames that actually led to nonsense-mediated decay. In this case, they found that the primary isoform resulted in insufficient MFN2 protein function, which explained the observed Charcot-Marie Tooth phenotype in this program. In the second paper, researchers at Radboud University developed an analysis pipeline to leverage long-read iso-seq data to help predict the functional impact of variants of unknown significance in rare disease cases, which is a common use case. And they tested their pipeline out on 5 unsolved probands and also found an increase in explanation rate for rare disease. So if you are only doing exomes or genomes for rare disease, hopefully I have convinced you that you might want to consider adding long-read RNA sequencing as well. In the next talk, you will see more examples of this and hear about how researchers at the University of Southampton and the NHS are using PacBio full-length Kinnex data to better understand short-read negative cases of rare disease.
Hi everyone, I'm Carolina. I'm a postdoc at the University of Southampton, and today I will be talking about our work using long-read RNA-seq to investigate splicing variants in rare disease pathogenesis. So for a bit of context, rare diseases are rare on their own, but in general, as a whole, they're actually there are over 7,000 rare diseases in general, and in the UK alone, they affect about 3.5 million people. The majority of rare diseases have a genetic component, and because of this, a lot of the new cases are found in children. But a stark figure is that only 5% of rare diseases actually have approved treatments. And in order for us to develop new treatments for rare diseases, we kind of have to be very sure that what is causing this condition. And one of the biggest bottlenecks that we have in genomic research is that we are producing lots of DNA sequencing data in order to help aid the diagnoses of rare disorders. But we have a huge bottleneck in the interpretation of this data. We know that whole exome and whole genome sequencing still leave a large percentage of rare disease patients undiagnosed. And this is evidenced, or was evidenced, in a pilot study from the UK's 100,000 Genomes project, which yielded a genetic diagnosis for about 25% of the probands. And to date, the figure still is roughly around between 20 and 25%. And it's different for different disorders. Some have a much higher rate than others, but in general, it's about this number. And this sort of bottleneck is quite clear when we look at databases like ClinVar. Some of you might know this is a free database where we have genotype to phenotype correlations, and the variants that are found within this database, almost half of them are variants of uncertain significance. And this amounts to over 1 million variants where we're not really sure whether they're pathogenic or benign. And having this really clear is going to be very important when we want to diagnose a patient. So we're in the clinic. We need to be certain what the variant does. And so there's a massive need to understand VUSs in order to improve solve rates in rare disorders. Here in Southampton, we are interested in looking at splicing in order to increase the solve rates in rare disease research. And splicing is the process in which the mRNA is spliced. And so in this case, the introns are taken out and exons are joined together to form a mature RNA. And splicing is controlled by lots of different elements. We could have cis-acting elements such as splice enhancers or silencers. We've got the canonical splice sites, the branch point, but we also have trans-acting elements that regulate splicing. And disruption of splicing on it as a whole isn't well studied, but we are really interested because we believe that we can uplift the rate of diagnostics by ascertaining and validating splicing variants. And here in Southampton, we've been working extensively with short-read RNA-seq in order to do this. And we have been developing a short-read pipeline in order to interpret and classify variants of uncertain significance that are likely to affect splicing, and also find pathogenic events where we don't have a variant of uncertain significance or a candidate gene that might be causing the disorder. Using RNA-seq to look at splicing could also help us find new genes that are linked to disease, and it also opens up new avenues for treatments such as the use of antisense oligonucleotides. And like I mentioned, our group has been really focused on using short-read RNA-seq, and we're actually now in the process of trying to see if we can integrate this within our health system, within the NHS. And we have some preliminary results from our pilot project, and now we're looking into long-read sequencing and how that could potentially complement what we've been doing already. And as I mentioned before, there's lots of different things that can regulate splicing, but a variant within any of these elements could cause aberrant splicing, and these are just some of the examples of what we could see. So here at the top, we've got what we would consider normal splicing, and then the mature RNA. These 3 introns are taken out, and we've got all 4 exons left. Sometimes a variant could cause exon skipping, and what happens is just that this entire bit gets spliced out, and our mature RNA ends up having only these 3 exons with a much more shorter transcript. We could also have alternative 5' and 3' splice sites. So in this case, they're within the exons, and the transcript that result from these are much smaller transcripts than our reference. You could also have some intron retention, where actually the intron isn't spliced out and is still left within, and so this causes an insertion. And we could also have mutually exclusive exons, and so we've got a transcript that just doesn't look anything like our reference. And so we could have any of these events that could be caused by a variant. So as I mentioned before, we have a short-read RNA-seq pipeline, and so we've got a large cohort of patients that we have collected as a collaboration across the UK. Clinicians have gotten in touch with us, and they say, we think these cases have a variant of uncertain significance that could be solved using RNA-seq, or sometimes they send cases where they don't have a VUS and we're trying to find anything out of there. And so from our cohort that we had already collected, and we had short read sequencing and RT-PCR, we selected the cases that we thought would be most useful if they had long read RNA-seq. And then we collected whole blood and packaging tubes for these patients. And because this is sort of our pilot project, we sent 9 samples that had enough material, high quality with high RIN scores. And then these 9 samples were sent over to Menlo Park, where they underwent the library prep with the Kinnex kits and sequencing as well. Just to note, these 9 samples were pooled. So we had 4 different pools. Pool 1, 2, and 3 were all 9 unique samples, and these all underwent globin depletion. And pool 4 is identical to pool 3, but it didn't have globin depletion, just because initially we weren't sure whether this was going to be a necessary step. When we look at short read sequencing, it tends to be just a natural step and it's usually done. But because of what that means in terms of time, cost, and potentially what that could do to the quality of the RNA, we just weren't sure. So we had this extra pool that didn't have globin depletion. And once the samples had been sequenced, they went through the iso-seq pipeline, and this was all done by PacBio. And this is just a schematic of that pipeline. And what I found really interesting about using PacBio is sort of these steps right here, where we look at where it has the clustering. Normally, we're not really used to kind of clustering our reads and having consensus reads. It's very important for us to kind of look at the depth and how much evidence we have supporting the different junctions. So this was a bit new to me, and I had to sort of kind of wrap my head around what I was actually looking at when I was looking at the BAM file, or at least this process BAM file that I got. And then once the reads were clustered and mapped, then they also they were classified. And I'll show a bit in the next slide about the different categories. And we end up getting a file that sort of looks like this, where we've got all of the transcripts, whether what categories they were in, and then the transcripts per million. And I'm just going to have here, this is the list of files that I actually got from PacBio, but I want to focus on these ones that I've got in red. So these are the files that kind of look like this. And we've got a classification file and then a filtered file. So there's a function where you can use to filter the results. And I wanted to look at these 2 in particular because I feel that these would be the ones that would be helpful in maybe looking at for the unknown cases that we had. And here I've just got the structural categories that we see that are in this annotated file. And so the top here, we've got the reference transcript. This one in blue is a full splice match. So all of the splice junctions match perfectly. We've got here an incomplete splice match. So there's a partial one that's missing this exon right here. We've got not in catalog. So novel not in catalog, or sorry, novel in catalog, not in catalog is the bottom one. So this novel in catalog is, we've got a new isoform and it's got the same junctions as our reference, but it's missing this exon. So it's a new combination of splice sites, but it's known splice sites. And the novel not in catalog is where we got completely new splice sites that just hadn't been seen within the reference. And so these were the main structural categories that are within these files. And so I thought it'd be useful to show how I've been looking at the data, because this is a pilot project. We've only just started looking at PacBio data, and this is sort of our first pass. We have samples that have known variants that we know are causing specific events, and so we wanted to look at those. And we also have cases where we don't know, and so that's going to be for later down the line once we actually come up with a plan of action on how we can pick out those novel events. But this is sort of how we would go about if we had just a sample that was sent to us and we would look at it using short read sequencing. It would be a very similar scenario. So for this particular case, we've got a UBR4 variant, and it's predicted to affect splicing by Splice AI. And this is the screenshot in IGV. This is the short read data. And we can see the variant right here in blue and what looks like to be intron retention. This is the sashimi plot for that particular case and some controls. And for those of you that aren't familiar with sashimi plots, they show us the coverage of the exons of the region, and so that's why the exons have a bit more coverage. And then these lines show us the junctions that we see within the BAM files. And so these are usually split reads. And with this particular case, I thought, well, this looks like there's likely an intron retention. But when we looked at the controls, you couldn't really tell whether that was just a normal event, or if it was just if it was a sample. So for this particular case, this is what the short reads look like. It's quite a long intron as well, so even using RT-PCR might be a bit difficult to do. And this is what the PacBio data looks like. So I've just zoomed in on the exons of interest. And so we see the variant right here, and then we see all of these transcripts that show intron retention. And there's another one transcript at the top here, where actually it's not intron retention, but we have a brand new donor site here. And then I went back to look at the file that I had mentioned before, so the classification file, but I'm looking at the filtered file, because just to give me an idea how many transcripts is this actually picking up per sample, because then for the ones that are unknown, I want to have an idea how much am I going to have to filter down in order to get to the pathogenic event. We have to remember that when we're looking at aberrant splicing, we're going to have normal events anyway that are going to pop up. So it's being able to determine what is a pathogenic event and what is normal. And so for this particular sample, we see that we've got over 6,000 full splice match for all transcripts. And again, this is a filter file. And we've got almost 95 incomplete splice match. And for the particular gene that I was interested in, this UBR4, this is what we had. So we had 64 incomplete splice match, 15 novel in catalog, 13 novel not in catalog, and 3 full splice match. And as I mentioned before, I made the mistake initially of thinking, this is a BAM file and this is the depth, but actually each one of these reads is a consensus reads, and they are backed up by more than just one read, that as you would assume here. And then when I looked at the specific UBR4 transcripts, these intron retention were found in this novel not in catalog, and so 12 out of the 13 are this intron retention event. The next case I wanted to show was this KLHL7. It's a homozygous deletion that is also predicted to affect splicing. And this is what the PacBio data looks like for that particular case. And we see that deletion here. And there seems to be a loss of that donor site. There is what seems like a normal junction here, which to me is a bit odd because this is a homozygous deletion. So we're looking into this of why that's there. And again, I did the same thing. So this definitely confirms that we're not seeing what we expect to see as normal splicing. And for this case, these transcripts were actually not in this filtered classification file. They were in the non-filtered file, and they were also as a novel not in catalog. So that gives me a good idea that probably I won't want to be using this filtered one down the line when I'm looking at the unknown cases. And I've got the same exact variant, but this time is heterozygous variant, and we can see very similar. We definitely see that loss of the donor site right here, but we do see about half of normal transcripts and what potentially looks like a exon skipping here. And again, similarly to the previous one, these particular isoforms were not in this filtered file, they were in the unfiltered file. This other case, this is an NF1, also a deletion variant, and it's also predicted to affect splicing, and it also leads to exon skipping as well. So this are what the Illumina short reads look like. So we can definitely see the variant here, and we see that some of them have the normal junction, so these right here, but there's also some exon skipping, and so here that's a bit clearer in the sashimi plot, where we see 6 reads that show the exon skipping, but only looking at the sashimi plot, we wouldn't be able to sort of tell that we also have some transcripts that are missing these 12 nucleotides that have the normal junction as well. Now, looking at the PacBio data, it's definitely a lot cleaner, and so we do see the exon skipping in these isoforms here, and we also see normal junctions with that deletion. And again, looking at the total number of transcripts that were identified, we had very similar numbers. So we have in the over 60,000 full splice match, over 100,000 incomplete splice match. And then when I looked at the specific NF1 transcripts, 2 of these new transcripts were in here, novel in catalog, but the rest weren't. So again, while this filter classification file might be useful sort of to kind of look at the bigger picture, when we're looking for such specific events, we probably want to use the unfiltered classification file. And this case I thought was really interesting as well. So we've got this COL9A2 variant, also predicted to affect splicing, and it's what we've seen in the RNA-seq is that it causes exon skipping. So this exon 30 is being skipped, and we can see that very clearly here in the PacBio data. But what we can also see is that over here upstream, we've got an intron retention, and initially I thought, is it potentially linked to the variant as well? But we saw it in other samples, so this seems like it's a normal event, it's just not annotated. But what I found more interesting is that this variant affects the normal transcript. So we see in this case, this transcript, sorry, we have the exon skipping and we don't have the intron retention, but we do have isoforms that have the intron retention and also have the exon skipping. So then this leads to really us being to ask a lot more questions as to whether, you know, how are these different isoforms, what is the effect that this is going to have on these isoforms, and how do they correlate to disease? And it's something that we really can't answer when we have short read data, because we won't have all of this information downstream. And sort of just to say as well that for all of the cases that I've shown here, we have confirmed the events by short read RNA-seq and RT-PCR. So that's sort of the first couple of things I wanted to talk about, just kind of give a sense of how looking at this data can help us with the identification of aberrant splicing events in these type of cases where we have a VUS that we think might affect splicing, and how long read can aid in that, in sort of covering the gaps that are left by short read RNA-seq. But then the other bit that I wanted to talk about was actually the comparison of globin depleted versus undepleted. And I think when I started working with RNA-seq data, it was, whenever I asked, it was just sort of, oh yes, you do globin depletion, but we didn't really kind of, I never really kind of asked too much, because I'm not a wet lab person, so I just kind of took the word that saying, you know, we need globin depletion. And what we found, or what I found when looking at these two pools where we had the exact same sample, one with globin depletion and one without the globin depletion, is actually the undepleted pool. All of the samples within that pool show a much higher number of transcripts in all categories. So the full splice match, incomplete splice match, novel in catalog, and not in catalog. So these are probably the most important ones. And we see that same pattern across all 4 samples. Here we see biosample 3 and 4. I'm not sure why biosample 4, the difference is much bigger. It could be a depth thing, but that's something to look into. And I had done this previously with short read data. So this is a plot where I've got the medium log TPM values for some batches that we had that were depleted, and then by accident we had a batch that wasn't depleted. It wasn't globin depleted, and so that's this batch 3. And this is the correlation of the TPM values across the transcriptome. And there's actually a pretty good correlation, and of course we see the globin genes sort of stick out here. But actually, the TPM values that we see in PacBio, for example, of these particular globin genes are comparable to what we see in the short read. So I think that was a really good sense check to actually, we're seeing things that are very similar with the short read data. And then I had also looked at specific genes. So these were all genes within our short read data that had a VUS in it. And so naturally we were interested to see, do we have enough coverage to actually analyze and look at these genes. And as it turns out, batch 3, which is the one in pink, and it was the one that did not have the globin depletion, had much higher TPM values than medium TPM values than the rest of the other batches. And I decided to kind of look a bit more at the genes that had a full splice or incomplete splice match. So these are unique genes. And when we look at the depleted versus the undepleted for all 4 samples, you do have about 1,000 more genes that show up with that you can identify in the undepleted versus the depleted. And then looking at GMS panel app genes, which are likely the ones that we will be looking at, because most of these VUSs will be in these genes. The difference is actually not huge. I've got the percentages here, so they are comparable. I think the difference would be in the depth and the coverage of these transcripts. We see that there's a lot more of these in the undepleted. While yes, we get more transcripts, they are likely just at a much lower depth. So they have much lower TPM values than the rest. And so that's sort of where you'd have to have a balance. You know, are you looking more diversity and slightly less coverage, or maybe a bit more coverage to pick up the genes that you're interested in, because obviously those globin genes do take up quite a lot of the reads. So in summary, I I hope that I've shown how long-read RNA-seq can aid in the identification of alternative splicing events, which are difficult to assess using short reads. In my opinion, the long-read RNA-seq is really, really useful when we're looking at intron retention. And just complex events in general, like I showed that case where downstream there was just a normal isoform with intron retention, but it's not something we would have been able to pick up with RNA with short read. We also have, I didn't show it here, but we have some long read data for another gene, the SF3B4 gene, which doesn't map very well with short reads. And with the long read data, we could see very well a new event. And so it could, just like with DNA, if it has a bad mapability, long read can definitely help in that regard. And something that I haven't done too much of, but I'm really hoping to kind of move on and do, is the quantification of isoforms and actually looking at how are these isoforms, what are the ratios, what are the downstream kind of pathways that are being affected. And you know, we can't really look at differential transcript usage with the short reads, whereas we can with this. So that's something that I'm hoping to look at in the future. Another thing that sort of is highlighted by the long read data is that the effects of splicing can actually be more complex than we expect. And that actually just pops up more questions. So how does this correlate with the disease mechanism? You know, we constantly get questions of how much splicing, how much aberrant splicing is needed to cause disease, and that's something that we're still trying to figure out. But of course, when we look at these events, they come with phenotype information, sometimes DNA information, and it's a combination of all of this data to really kind of pinpoint what's happening and what's going wrong. And something that would also be really, really useful and we'd love to do is look at other tissues and say, do we see the same events, the same patterns in other tissues, the same isoforms? And with regards to the globin depletion, the results that we got with the long read data are actually very similar to what we had observed previously in the short read data. The undepleted samples definitely had more splice match transcripts as well as incomplete splice match. So definitely was identifying a much higher diversity of transcripts. The undepleted samples had a higher number of uniquely genes expressed. But then again, if we focus on this GMSF panel lat genes, the results are comparable. So that really depends on sort of what you're interested in. I think in our opinion, we decided for future cases, we probably weren't going to do globin depletion just because unbalanced time, costs, we didn't think it was worth it. And that's it for me today. So thank you, and thank you to everyone here at the university, and of course all the PacBio team that has helped kind of get this through. And I will answer any questions.
Thank you, Carolina, for an excellent talk. Very exciting work, and we do have a number of questions. I'm trying to organize them by topic, but it might be kind of all over the place to start with. Maybe I'll start with a general question for you, Carolina. What was your experience, and do you have any lessons learned in switching from short read RNA-seq analysis to long read? Was it difficult? Do you have any recommendations? Were you able to just apply your same pipeline, or did you have to make significant modifications?
So, I mean, in terms of switching from short read to long read, it was because of, so the way that we tend to look at splicing, we have a variant that we're interested in, and we go straight to that gene, that isoform, and you know, we go into the BAM file and can we see something? And so I took that same approach for the VUSs. For the unknown cases where we don't actually have a VUS, I'm still trying to determine how to apply sort of the pipeline that we use to pick up some new events, and that might be related to a pathogenic variant. I think the only thing that I found was that with the short reads, you still get a lot of the intronic variants showing up that are causing the aberrant splicing, whereas with the long reads, they don't tend, they get spliced out. So that was one thing that's slightly different, and that I might have to modify my pipeline, because I would do some variant calling on the short read RNA-seq and then overlap that with the alternative splicing that I found. And so I think that's something I'll have to modify for it. But I think so far it's been relatively sort of simple, kind of switching from one to the other. And I think, like I mentioned in the talk, having the clustered isoforms, I think that was different for me, and I think I probably will have to go back to the unclustered isoforms and see how to integrate that into the pipeline.
Great, thank you. Maybe let's see a question for Liz about the iso-seq pipeline that Carolina used. You mentioned, filtered, unfiltered. What's the difference there?
Yes. So this is based on SmartLink's Pigeon, which is actually a PacBio's implementation of a tool called Scanti. You can look at Scanti 3, so SQANTI 3. It's in its third version. It's an isoform classification and filtering system. And the filtering are, of course, there's always heuristics, right? And so, Carolina, you found that there might be instances where the filtering was overly aggressive, but by and large, it's trying to filter out the following: intrapriming artifacts, so ones where there are A's is part of the transcript, and the poly to T priming in the process of generating the cDNA might actually prime off that. So that's actually a false end site, and so that could be bioinformatically filtered out. Another is potentially template switching artifacts. This was described in the very first Scanti paper as a common library artifact from cDNA generation. So there are certain patterns you can also bioinformatically detect and remove them. Other potential artifacts that commonly get removed are unspliced, single exon, intragenic, otherwise unannotated transcripts that don't have any supporting evidence. So these are described in the original Scanti paper. The new Scanti 3 preprint has more refined machine learning based filtering that sometimes will actually rescue some of these overly aggressively filtered transcripts. I know that's a long-winded answer.
No, thanks. And I think that probably addresses the question about artifacts and false discoveries as well.
Yeah.
Okay, great. Let's maybe, for Carolina, there are a few questions about some of the findings you have. In general, for splicing-related variants that were being shown, were findings different for those with variants as compared to other samples lacking those variants in regard to intron retention, exon skipping, etc.? I don't know if that makes sense.
So how did the ones with the variants compare to
I think
my
interpretation of that question, because I was also looking at it, was that, Carolina, you have pointed out there's intron retention that was unannotated. And I think what they're asking is, like, do you also see exon skipping events and intron retention that were novel in non-variant genes, like genes that were not what you're targeting?
Yes, 100%. I found that there was quite a lot of novel isoforms that were not associated with pathogenic variants, but they were regardless still showing up. And so that's why I put that example there, just to show that this is a completely new event, but that was also found in with the normal samples, that just wasn't related to this particular pathogenic variant. But yes, I guess that would be the answer.
Somebody was asking specifically about the UVR example. It said there were reads that included multiple introns. Were those reads expected and represent transcripts prior to mRNA splicing, or were those also pathogenic events?
So I think those particular ones were artifacts, and when you looked at that filtering and that Liz was talking about, they get taken out. So these are more likely artifacts, yeah.
I had a question in general about, you mentioned at the end the GMS panel-app genes. For those outside the UK genomics community, can you talk a bit more about Genomic Medicine Service Panel app? And then I think with interpretation, how does that help you having a targeted list to look at?
Yes, so within the genomics service here in the UK, if for the most part the way that genomic data is analysed, if a patient comes in with a specific disorder linked to a phenotype, whether they get whole genome sequencing, we don't tend to look at the entire genome. That whole sequencing, the whole genome will get filtered down based on panels. And so these have been experts that have sat down and said for, I don't know, developmental disorders, we've got this panel of genes that we look at within the NHS. And so the way that they go through, it's, you know, you have the whole genome sequencing for that particular disorder, but in the end, we actually only tend to look at what's in these panels. And so what's more likely to get sent to us as a VUS is going to be within these genes within these panels. We probably won't tend to get too many things outside of it unless it's one of those cases where we just have no idea what it is and we might be looking at new disease genes. But so that list really helps us sort of narrow down what are the most likely genes that we will be assessing using the RNA-seq data. And so that's why that's important, because we're very much likely going to focus on that list of genes. It's about, I want to say it's about 4,000 genes at the moment. It gets updated every so often.
Great. There are a number of questions around globin depletion. So maybe high level, you show difference in number of isoforms between depleted and non-depleted. Do you think that globin depletion is non-specifically removing other isoforms, or is this just like a low sample size that we're looking at differences? And is the quality of the data for the batch without globin depletion still good?
Yeah, so the quality of the data is still good. But as I mentioned, there's going to be quite a lot of reads taken up with the globin genes. And I'm not exactly sure how the globin depletion is being done, if it's an enzymatic depletion, because it could be taking out some stuff that some transcripts that we might be interested in as well. So we recently had a discussion about the globin depletion as well, whether it's taking out some things that we might be interested in. I think for sure it's definitely taking out some things as well. But it's always a discussion that we have with, you know, do we do it, do we not? And from what I've seen, the way that I've been looking at the data, having globin depletion didn't really make a huge difference.
I think I wanted to, you know, maybe add a little bit because I could see people were very, very interested in this question, right? And Carolina, I had helped a bit and look at the stats. I want to point out two things. One is the sequencing depth were slightly variable. I think it was in one of your slides, right? The number of reads that we had gotten for the 4 samples was slightly variable. And it was interesting that the one where we got the most number of transcripts in the undepleted sample was also the one that had the highest number of sequencing depth. And I almost felt like, you know, even we could, if we want to go down the road of like normalizing the read depth, we could, but it almost indicated to me that the effort, at least in this case, the effort it would take to do the depletion could be compensated by just doing a little better on sequencing. It was almost that when I was like, if you just sequenced a bit more, and the sequencing depth on all of them were kind of comparable. It was just like slightly better day versus a better day. And it's like, if you just loaded a little better that day, you didn't have to do the depletion, at least in this very particular case. And it is the depletion that was used was a CRISPR-based depletion method. I think, you know, for curiosity, we could go and look at which genes and isoforms were depleted, and was it, you know, accidental depletion because of similarities.
There was a question, do you know, Liz, if RIN or transcript size was affected with depletion? or anything?
Rin, I don't know. That's a good question. But the size, my feeling was no, and the reason is there were very few. The most dominantly depleted were just the globin genes. The other ones, we should probably look at whether they're low abundance, and maybe that's why we're really not seeing a lot of major changes.
Just one last question on that. Blah, blah. What's the rationale for higher TPM for non-hemoglobin transcripts in the non-depleted samples?
Oh, what's one of your slides? So I'm trying to remember what's that there. So I think, Carolina, you had a slide that showed the TPM.
Or when we looked at the genes that we were interested in. Yes, I'm going to, sorry, I think I'm just gonna quickly turn off my webcam because my internet is very slow right now. And I'll share. I think it was this one. I don't know, I think we were just as baffled as well by the fact that we also had higher TPMs within these genes, and I think it's just, you know, maybe the globin depletion does have some off targets within certain genes, but you know, I haven't really kind of quantified and really looked at exactly what is happening with those other genes.
Okay. Okay, we just have a few more minutes. We have a bunch of Kinnex questions, so maybe I'll pose these to Liz, or maybe Carolina. There's a question about using whole blood as informative for some of these diseases, because some of them might be tissue specific. Some of the genes might be tissue specific with regards to expression. Can PacBio plus Kinnex be made compatible with low amount of biopsies available from patients? And they're asking about CNS diseases, but maybe just in general, tissue specific capture.
Yeah. Well, I think it's talking about potential low input, right, or low incidence scores. So, you know, currently the Kinnex kit uses the iso-seq Express 2.0 kit to generate cDNA from total RNA. Our standard is a total RNA of 300 nanograms and RIN over 7. It's not that you can't use lower RIN samples, but it almost certainly means lower RIN will result in shorter transcripts, lower quality. If the RIN score is really low, you might not actually get very long transcript, and that defeats the purpose of doing long read sequencing in the first place. As to the desire to have less RNA amount, I don't think we've tried total RNA significantly less than 300 nanograms. I would be interested from the people who are asking these questions, generally, what is the total RNA amount and the RIN score that you're seeing, because there could be other methods that we could use to, still get good results. We just haven't tried it.
General questions about, you mentioned low abundance transcripts. Can you talk about how low we can detect and where they can find more info?
I can say theoretically, based on our, data depth, but I'm also curious if Carolina had looked at any low depth genes for your research. But for us, it's really just looking at the total number of reads, right? So more sequencing means you capture more things. The throughput right now for Kinnex on Revio is 40 million full-length cDNA molecules after this concatenation, deconcatenation. So if you have something that's expressed, at 1 transcript per million, then on average you should be expecting, 40 read counts. And in the long read world, 40 reads is a lot. In general, anything above 2 to 3 reads is quite sufficient evidence.
Okay. Question about, do you lose any transcripts due to concatenations may be longer isoforms. I feel like we get this every question.
Yeah, so the answer is no, and then this is actually in our app note. And I actually want to leave the last minute because I saw an excellent question that has just come in for Carolina that I thought would be wonderful to get her thought on it before we end this call.
Go ahead, Liz, you can.
Oh, so it just came in, and Carolina, she actually the person asking, you know, your team's experience using the new Revio and how you plan to use it beyond RNA, genetic diseases, and other non-human samples at your institution.
Sorry, could you repeat it? My internet is cutting off.
Okay, so it's generally like, what's your general impression of having used this data so far? What is your institution's plans for just going beyond Kinnex, you know, or even for non-human, if that's at all something that you guys work on?
So we're mostly focused on human. So at the moment, we don't have any plans, but we do have a collaboration with the Xenopus Research Centre in Portsmouth. So there might be potentially in the future looking at some frog RNA. But I was, you know, my first impressions of using this data set is that it was really the fact, as a bioinformatician, the data itself was really clean, and the size of the data just was a huge advantage because that saved computational time and resources when we're actually processing all of this. I think that was one of the biggest things that I found I really liked about this data set. I've worked with some ONT data before, and it's really, really large, and it took me weeks to just process one sample. And so with the PacBio data, the fact that it was so much smaller, such high quality, I think we're really interested in continuing to having a much bigger data set and more samples being sequenced as well to really have a comprehensive view of how we can continue to use this within our research and potentially within the NHS.
Perfect. Thank you. So with that, I guess we will wrap up. Thanks for attending.